today's speaker. And we are now recording. And so it gives me a great deal of pleasure to introduce one of my closest colleagues and friends, Richard Kuhn, who is the Trent and Judith Anderson Distinguished Professor of Science in the Markey Center for Structural Biology at Purdue University in the Department of Biological Sciences. Richard received his PhD in 1986 in molecular virology from Stony Brook. And that's where I first met Richard, in Eckhard Wimmer's lab, where he was just a beginning graduate student and I was a postdoctoral fellow. And he was really a geeky New York science nerd who looked like he was about 16 years old. And in fact, he looked so young at the time that one of our lab mates in the Wimmer lab nicknamed him Rich Boy. But Richard very quickly found his way to the adult scientific table, where he displayed his incredible smarts, his skills as an experimentalist, and his strong professional drive. During his thesis research on the molecular genetics of picornaviruses, Richard was first author or co-author on 15 publications, an amazing PhD output. In addition to his groundbreaking studies on the genome-linked protein of poliovirus, Richard had time to build collaborations and friendships with me, for example, that have lasted 40 years. We also had many opportunities to share late-night beers in the Wimmer lab, where the only rule was that we couldn't start an experiment after midnight. Beyond our shared fondness for science and alcoholic beverages, Richard and I also shared a love of rock music, especially the Beatles and the Doors, further cementing our almost lifelong friendship. Richard left Stony Brook to pursue his postdoctoral research in Jim Strauss's lab at Caltech, where he carried out some of the first reverse genetics research on alpha virus like Synbus virus. It was also a chance for us to get together scientifically and socially here in Southern California. My wife Ronnie and I were lucky enough to be present with Richard and his wife Charlene for the christening of their firstborn child, although I wasn't sure if a little boy would survive being in a Catholic church with a non-believer like me. After his postdoc, Richard assumed an assistant professor position at Purdue, where he expanded his research on alpha viruses to uncover the molecular determinants of virus replication, assembly, and virus budding. In addition, he began a decades-long collaboration with the late Michael Rossman to investigate the structure-function relationships of a range of RNA viruses, including alpha viruses and flaviviruses. It was truly a synergistic collaboration that led to a large number of significant discoveries and dozens of high-profile papers in journals like Science, PNAS, Cell, Journal of Virology, EMBO Journal, Nature, you name them all. Indeed, Richard has published more than 200 papers during his academic career. So not surprisingly, he is a very sought-after seminar and meeting speaker. By my count, he's given more than 150 invited presentations worldwide during the last 10 years alone. And that includes the travel restrictions from last year due to COVID. So you can do the math on the frequent flyer miles, but you can bet that he never flies coach on airplanes. In addition, his research program at Purdue is very well funded by multiple multiple grants from NIH. Richard has had numerous awards and honors, including an NIH National Research Service Award, an undergraduate teaching award. He had a named lectureship at the Karolinska Institute. He's an elected fellow of the American Academy of Microbiology, as well as an elected fellow of AAAS. He's won the McCoy Lecture for Outstanding Research Contribution, and he was the ASM named lecturer. Beyond his scientific prowess, Richard is a dedicated academic citizen. At Purdue, where he was promoted to professor in 2002, he has served as department head of a large biology department for 12 years. He was director of the Binley Bioscience Center for 9 years, and is currently the Krenicki Family Director in Inflammation, Immunology, and Infectious Disease at Purdue. And beyond Purdue, he's made major contributions in editorial, advisory, and review boards, including being on the editorial board for virology for almost 20 years, where he's now, in fact, the editor-in-chief. He's on the editorial board for microbiology and molecular biology reviews, where he, up until recently, worked for the editor-in-chief, Michael Bookmeyer. And he's on the editorial board for the Journal of Virology, where he continues to work for the editor-in-chief, Roz Sandry-Golden. He's also been on the editorial board for viruses and PLOS pathogens as an associate editor. He's been very active in reviewing grants, including 2 terms of as charter member of an NIH study section, and more ad hoc reviews than you can imagine. I couldn't even keep track of all of them on this. So without further ado, and so I don't want to take up any more of Richard's valuable lecture time, I want you to join me in welcoming him for his seminar entitled Structural Studies of Zika, Dengue, and Related Viruses. Richard?
Wow, that was an incredible introduction, Bert. Certainly, you could have shortened it by about 10 minutes. But I appreciate the kind words, and it's really good to be back talking with a lot of my old friends and colleagues and new ones, which I'll talk to in a little bit. I do want to say, given that introduction, Bert, that I tell prospective graduate students that they may know what they want to work on, but it's more important to think about the lab environment that they are going to spend the next 4 or 5 years in. And that was certainly true for me, because you may not remember, but I wasn't interested in viruses when I rotated in Eckhart's lab. I was interested in neurobiology. And it was really the interaction of Bert and Ronnie and a number of other colleagues that kind of changed my view of where I wanted to be and how I wanted to spend the next few years as a PhD. So those interactions obviously have changed my life, my career, and long-term friendships. And I want to thank you, Bert, for those early days and years. So with that, I will start, if I can find where my talk is. Oh, it's right here. And uh-oh, switch. Sorry about that. Okay, now that's better. All right, so as Bert said, because of my multiple monitors. All right, I'm going to have to switch. We didn't check that one, Bert.
Will the power grid in Indiana support that?
We're okay in Indiana. No, it's just switching between monitors. So what I'd like to do is go over a period of about 20 years of research, bringing you up to date on the most recent work that we are actually submitting for publication this week. I'll start off by saying that we solved the structure using cryo-electron microscopy of dengue, which is a member of the flavivirus genus of Flaviviridae, 20 years ago. And at that point, and I'll show you those early images, we thought we really understood what a mature flavivirus looked like. But it's not true. We had a lot to learn, and we've learned quite a bit, and we still have more to learn in the next few years. What we found is that there is significant translational and conformational changes that occur during the maturation of the virus. And as the viruses we call escape from maturation. The viruses, at least the dengue viruses in particular, never truly grow up. And I'll explain what I mean by that in a little bit. It's also true that although we look at these viruses and we have this static picture of a nice spherical, pretty looking particle, turns out that these viruses, particularly the flaviviruses, are very dynamic. And that plays a role in antibody binding, probably the immune response to the viruses, and parts of the biology of the viruses. Temperature also plays a role in this dynamic process. And so I'll show you some early studies that we did that demonstrate that although the mature virus is a nice, smooth particle, at 37 degrees, it does not have the same structure. And more recent studies, we've demonstrated that these particles, although we call them icosahedral, may not be perfect icosahedral particles. They may be defective, and I'll suggest a reason, or at least a hypothesis, why that might be so. And then finally, at the very end, I'll throw in some of our antibody work that I think is interesting and relates to all of the topics that I'm going to talk about now. So my lab has been interested in correlating this plasticity, this heterogeneity in virus structure with the potential for influencing the tropism of the virus and the immune response and the disease of the virus. So this has been a kind of a long-term overriding theme in the laboratory. So just to bring everybody up to speed, flaviviruses are positive strand RNA viruses. They have a 5' cap, but the 3' end is not polyadenylated. They are about 50 nanometers or 500 angstroms in diameter. They're spherical, icosahedral, as I've already said, and they have a lipid bilayer membrane. They are distributed throughout the world, and they are transmitted primarily by insects, either ticks or mosquitoes. And there are more than 70 members. Many of these are associated with pretty significant human disease, dengue being at the top of that list because it is so pervasive in many areas of the world. But yellow fever, a virus in which we've had a great vaccine for more than 90 years, yellow fever still kills people in certain areas of the world. And then, of course, we're more familiar with West Nile and Zika viruses. These viruses cause a very broad spectrum of disease in humans. A lot of them, probably 70 to 80%, are asymptomatic infections. But if you feel that you're sick, you may simply have nausea or vomiting, the kind of classic symptoms of a virus infection. But you can go on to have encephalitis, or you could even have shock and hemorrhagic fever with dengue, for example. There are 3 genera within the Flaviviridae family. Hepatitis C is another one of these genera. We also work on hepatitis C, but I'm not going to talk about that today. This is just showing the distribution of mosquitoes in the world and levels of virus. Dengue is said to be the most significant arbovirus in the world, affecting nearly 400 people every year, with about 50 million cases that are clearly identified. And there are several 100,000 cases of a more severe form of the disease called dengue hemorrhagic fever. And I'm not going to talk about the difference in these 2 diseases. It's really a continuum of a single disease. There are 4 different serotypes of dengue virus. And so as defined by serotype, if you're infected with one of these serotypes, you're not protected against infection with another. And that leads to the complication of antibody enhancement and this more severe form of the disease. So its genome is pretty simple. It's 11,000 nucleotides in length. We're going to be interested in the very 5' end of the genome, which encodes for 3 structural proteins: the capsid protein, the membrane protein, or in its initial immature form, it's called the pre-membrane protein, or PRM, and then the envelope protein, which is the surface protein that really occupies just about the entire surface of the virus particle, as I'll show you in a minute. And these virus particles have icosahedral symmetry. This is just showing a genome map of the flaviviruses with our 3 structural proteins found at the amino-terminal end of a polyprotein that is going to be cleaved by a viral and cellular proteases into 10 individual proteins, the 3 proteins I've mentioned, and then 7 replication proteins that are involved in interacting with the host, antagonizing the innate response, and replicating the viral genome. This is the original structure that we did back about 20 years ago of dengue virus. And surprisingly, unlike most other envelope viruses, dengue virus, when we solved the structure, doesn't have projecting spikes. In fact, the envelope protein, which was first solved by crystallography by Felix Ray, Steve Harrison, and Franz Heinz back in 1995, and it was predicted at that time that that protein, based on antibody reactivity, may lay
parallel to the viral membrane and therefore lay flat. And that's exactly what we saw when we determined the structure using cryo-electron microscopy. Now, it's not apparent from this to most people, but this virus particle has icosahedral symmetry, and I've highlighted here the asymmetric unit, which I'm going to talk about a little bit, not in too much detail, but just enough to tease you a little bit. Of course, if it's a repeating icosahedral organization, if we understand what's inside of this asymmetric unit, we can use symmetry of an icosahedron to figure out the structure of the entire virus. This virus, we normally kind of referred to as a golf ball because it has that kind of surface feature, slightly dimpled, but no major projections. This blue surface view that you're looking at is entirely the E protein packed very tightly against one another. And I'm going to show that in the next few slides. Since the structure of this is dengue virus by cryo-EM, but the structure from Steve Harrison and Felix Ray was an X-ray crystal structure of the ectodomain of the envelope protein from tick-borne encephalitis virus, a related flavivirus. And what my colleague, Michael Rossman, and I and colleagues were able to do was to take the X-ray density that they had and fit it or dock it into the cryo-EM density that we obtained when we did this structure of dengue. And so what you see in 3 colors is the outline of the envelope protein or the ectodomain of the envelope protein, and it's how it's arranged on the surface of the virus. So it is found as a dimer, a crystallographic dimer, and a biological dimer in this mature virus. You can see that these 2 monomers of the E protein are related by symmetry, a 2-fold axis of symmetry. And so these were placed on the 2-fold axis of this asymmetric unit. And you can also see, I think, and I'm going to show it in the next slide where it'll be clearer, that these 3 molecules that we fit, red, blue, green, within this triangle are actually in different positions relative to one another. So we have 3 molecules that are identical, but they're in slightly different positions. And this can be seen here. This is now the C alpha backbone with all of the E proteins fitted into the dengue structure. It's color coded in that domain 1. We'll just say that this E protein has 3 domains. Domain 1 is in red, domain 2 is in yellow, and domain 3, which is an immunoglobulin-like domain, is in blue. And what I want to point out to illustrate this idea that these are non-equivalent positions, they're quasi-equivalent positions, is that the domain 3, the blue domain, here at the 5-fold, is very tightly packed. It's less tightly packed if we look here at the 3-fold, and then molecules that sit here on this 2-fold, domain 3 is got a lot of space between adjacent domain 3s. I'm going to come back to that concept when I show you just a little bit about antibodies. But it does have consequences for the biology of the virus in that even though it's the same coding sequence, it's the same protein, it really depends upon what position within this repeating unit it's actually found in. This is just the cross-section of our EM density. This is actually for Zika now, showing the inner core, which is RNA and capsid protein. Then we clearly can see a lipid bilayer. The inner leaflet of the bilayer is shown here in aqua, and then the outer leaflet is shown in green. The E protein and a very small M protein is shown here, mostly in yellow, but we can see both E and M transmembrane domains as they cross the bilayer. Importantly and interestingly, these transmembrane domains for E and M don't penetrate across or into the inside of the particle. They stop at the inner leaflet of the bilayer. And so when we look at this, there's no connection between the glycoproteins and the inner nucleocapsid core. And this is different than what we clearly have shown many years ago for the alpha viruses, in which each glycoprotein makes contact with each capsid protein. So there's a question right away in terms of how do you assemble a particle like this, and we'll get back to this in a bit. So this is just showing how the virus particle is matured. Viruses are made, they bud from the lumen into the lumen of the ER. The RNA is synthesized in vesicles within the cytoplasm. The capsid is in the cytoplasm. It associates with RNA, and you bud these particles that are immature. And I'm going to show you, we have structures of these immature virus particles. And unlike the smooth structure that I showed earlier, these have spikes on. And what will happen, and I'm not going into the details of that, is that as the immature spiky virus particle, which has, instead of M, it has the precursor, it has PRM, and PRM is this little red knob at the top, and it actually protects the fusion loop of the E protein from undergoing premature fusion, because what's going to happen, as with all proteins that are moving through the secretory pathway, as the virus encounters the trans-Golgi network, it's going to encounter an acidic pH. And in that acidic pH, what's going to happen is that these proteins are going to rearrange and they're going to fold down into that relatively smooth-like structure with PRM still attached. When they fold down, the furin cleavage site, which is the host protease, which is involved in the maturation of the virus, it now can recognize the cleavage site. In this immature spiky structure, the cleavage site is on the interior of the spike and can't be accessed. But when it moves into this position, it can now be accessed, and it can be cleaved by furin. But because it's still in an acidic environment, the PR domain remains attached until the virus particle is released into a neutral pH environment, probably when it exits into the extracellular milieu. And so this is just a diagram of the PRM protein. The PR portion, which gets cleaved by furin, as I just said, is the larger portion of the protein. And it's believed that one of its main functions, but probably not the only function, is to protect the particle as it's moving through a low pH environment. I should point out that the PR protein has a glycosylation site, and it is glycosylated. I should also say that this immature virus particle is non-infectious, whereas the cleaved product is infectious. We don't know what the intermediates are in terms of infectivity. This is showing some work that we did with Zika virus, showing now mature Zika on the top, ABC, and immature Zika virus, DEF. We have in the A and D, we have the surface shaded view of the virus, and we can clearly see how smooth the surface is in the mature virus, and we can see these projections, these spike-like projections coming off in the immature virus. More easily seen in this cross-section where you can see these projections coming off. And although it's not absolutely true, what I'm going to say, but the blue, this dark blue, is mostly the PR component. And the reason why I'm saying it's mostly true is that this is actually a radial colored distribution. But the PRM, the PR moiety of PRM is sitting right at the distal tip of this spike projection. And then this is just the C alpha of both of them. And I should just go back and say that with Zika virus, and this was back in 2016, this is the first time using new technologies with cryo-electron microscopy that we were able to do a cryo-EM structure and reconstruction in which we could actually build an atomic model straight from the cryo-EM density. So we didn't have to bother with any kind of crystal structure. We used the cryo-EM density straight away. This is just now views of the E and PRM protein. This is on the top, A and B is the immature structure looking from the side. and this is looking down through the center of one of these spikes. These spikes are not threefold related, but there are three molecules of E and PRM that form this spike-like projection. And then this is, in C, is the structure of E and M as shown in the mature virus particle. A side view, this is a view in which we have the C alpha backbone, and then this is looking down, straight down from the top at the E protein dimer. Okay, so I'm going to take you back a few years, and we had started in collaboration, and I should say that the bulk of the work, but not all of it, has been done with my colleague Michael Rossman, who I collaborated with for more than 25 years, and as Bert, I think, mentioned, passed away in 2019. We were interested in looking how antibodies, in this case, it was an antibody, a neutralizing monoclonal antibody, how it bound to dengue virus. And what we did is we took dengue virus and we took a FAB fragment from this monoclonal antibody 182, and we complexed it at room temperature and then did a cryo-EM structure. And when we did the structure, we found no change, no Fab attached. We found just a native virus particle, and we were perplexed. We had colleagues at Washington University, David Fremont and Michael Diamond, who had demonstrated using crystallography of just part of the E protein and the Fab. The binding site for this Fab should not be accessible in this structure of the mature virus that we had solved. And so that was a bit of a conundrum. But of course, neutralization assays are not done at room temperature. They're done usually at 37 degrees. And so what we did was we incubated this complex at 37 degrees and then froze it and then did a reconstruction. And when we did that, we found that indeed the Fab fragment did bind and that the virus particle had expanded, and that cryptic site that was not accessible in the native structure that we had was now accessible because the virus had expanded out and that site became accessible. Presumably it occurred in a very localized region, and then you got a Fab to bind to it, and then you got spreading around that. And so you push the equilibrium such that the entire particle, for the most part, had FAB bound to this site. And so what that demonstrated to us, that even though we have this beautiful golf ball dengue virus, that's probably not the true only state virus, that the virus is actually breathing, which was shown previously with a number of other viruses, including coronaviruses. And so this is an example of an envelope virus, which is breathing. Excuse me. And the and the FAB can capture that stuff. Now, another experiment that we did, sorry about my throat, is that we took the virus, normally we do it at room temperature, and we incubate it at 37. And what we found is that the virus expands. Demonstrating the dynamic properties of the virus. When we look back at our early work on dengue, we found that looking at the particles, the raw particles, there were lots of different particles in there, lots of different morphologies. Some were smooth. The smooth ones we figured were good, but there were a lot of other ones that are represented here and here that looked to us at that time to be damaged. And so we didn't bother with them when we did those reconstructions. We threw out those particles. Well, it turns out that those particles represent different stages of maturation. And in the case of dengue 2, which is the first of those dengues that we did, a large fraction of those particles don't have PRM cleaved, leading to particles that are partially mature, or in fact, immature. Now, is this important? First of all, let me just summarize a few points here. There is a continuum of particles when we look in the dengue viruses in terms of maturation. Hence, this is what I said before about some particles never grow up. They never mature. But PRM cleavage is required for particles to be infectious. But we don't know, in the case of partially cleaved particles, whether these are as infectious as the mature virus. The presence of PRM is going to influence the interaction of particles with receptors and antibodies. In fact, antibodies against PRM have neutralizing antibodies. I'm going to show you some data at the very end and talk about that. And it's true that patients who experience secondary infection with the flaviviruses have elevated levels against the PRM protein. Hence, there's been a suggestion that the presence of PRM on dengue viruses may be related to the severity of disease. We decided that we would look at this using both cryo-electron microscopy as well as looking by mass spectrometry. Sorry. There we go. That's so easy to go back. There we go. So we use two methods. The one I've been talking about, cryo-electron microscopy, and another method called selected reaction monitoring, in which we quantified in a fairly rigorous way the amount of PRM relative to E protein and relative to M. This is just cryo-EM of a variety of flaviviruses. Kuhn is a West Nile type variant. Langet is a tick virus, and we all know about Zika. The common thread amongst these three viruses is that the particles look pretty homogeneous. There's not a lot of variation. Some of them are not perfectly round, but there's not like perturbations, protrusions coming off of them, unlike what you see for dengue 2 and to a lesser extent with dengue 4. This is just a blow up of that. Dengue 2 appears to be more immature than even dengue 4. And if you box these particles out in cryo-EM and you kind of put them in bins as to whether they are mature, that is perfectly smooth, whether they are perfectly spiky, that would be immature, or something in between, you find that dengue 2 is more immature, it has less cleavage of PRM than dengue 4. And when we looked by mass spectrometry, we found the same thing was true, that dengue 4, probably greater than 80% of particles had PRM cleaved, whereas when we looked in the dengue 2 serotype, in some cases, less than 50% of the PRM was cleaved. So this is all relating to this structural heterogeneity. I just talked about the PRM content. I coughed my way through the bumpy part that we observed at different temperatures. And then there's that antibody work that demonstrated breathing. And I'm not going to talk about the subviral particle contribution. But one might ask, what's going on with that cleavage? Why is it so inefficient, given that it's an RNA virus? This is a consensus cleavage site for human furin. And what I'd like to point your attention to is the P3, which is usually a basic residue. If we look in the flaviviruses at the P3 position, the flaviviruses don't have a basic residue. They usually have a neutral residue, or if we're looking in the dengue, the 4 dengue viruses, they're not even close. They have an acidic residue at that position. That is probably part of the reason why furin is so inefficient at cleaving. But one must ask, if we can mutate that site, why doesn't the virus mutate that site? In vitro experiments in which you mutate that site produce a virus particle that is not as infectious as the wild type virus. Now, the major question is, does the virus maintain this inefficient cleavage for its biological role in infecting host? And so we've suggested that this PRM content influences the structure and the dynamics of the virus particle. That clearly has an impact on infectivity and possibly the entry and exit from cells. This also will likely influence the immune response and ultimately pathogenesis. Now I'm going to change and talk about some newer experiments that identified some deviations from our perfect world of icosahedral symmetry. So I've already told you that we go from an immature spiky particle to a mature virus particle. And back when my student, Matt Thurkelson, wanted to do this experiment, the question was, what is the path by which these proteins move and rearrange to form this? And so what Matt thought he would do was to use an FAB, an antibody fragment, to try to trap an intermediate. And so he picked an antibody that we have called E16. This is against West Nile virus. He used Kuhn because it's easier. We don't need the BSL-3 to do the cryo experiments with. Kuhn is an attenuated relative of West Nile, but is still neutralized by this very potent antibody that Mike Diamond had originally produced. And if you take E16 and you try to add it to an immature virus particle, it should not bind because the binding site is not present in this orientation of the E protein. Fab16 binds to domain 3 of the E protein. So what Matt figured is we can change the pH and bring it from an immature to a mature virus particle and keep the antibody in there, the Fab fragment, and it will trap as soon as that surface becomes accessible. And we'll have a snapshot in between immature and mature. Nothing works the way we planned it. Okay, so here's, these are the classes of particles that we see from cryo-EM. This is the immature Kuhn virus. This is a virus, this is dengue virus with an antibody against PRM. And I'll show you later that we know exactly where this binds. There's 180 binding sites of PRM on this. It's well decorated. This is what it looks like by this 2D class average. When Matt did the same experiment with Kuhn in complex with E16, no pH change, in fact, here. This is just native with normal pH. He found that it looked like the original immature Kuhn, but there was some distortion occurring over on one side of the virus particle. And so what Matt did, which was, Matt was in my lab. This was not what we normally did, and certainly not in Michael's lab, was he did an asymmetric reconstruction of this virus particle. We do symmetric reconstructions because we can use the power of symmetry to improve our resolution. And so that's what we always did. So if you take these virus particles and you have these 2D classes, then you go through a symmetric reconstruction based on icosahedral symmetry, and you get a very nice particle. But if you did it Matt's way, you don't impose this symmetry on the particle. And what you find is, 4/5 of the particle looks great, but then there's something going on at the bottom. There's no Fab in here. This is just virus itself. And so if you do the reconstruction, this is the reconstruction of the immature virus using the icosahedral symmetry operators. Here is a reconstruction in which you don't impose icosahedral symmetry. What you see is that the core density of the capsid is pressed up against one side, which we're now going to call the proximal side or proximal pole. And on the opposite side, there's really a distortion and a lack of density that suggests that we're missing components, the bilayer and the glycoproteins, okay? I'm going to show you this. This is just a larger blow up. We actually, and I should have mentioned this earlier, in the immature Zika, we can actually see immature, not mature. We can see a connection between the core and the glycoproteins. And we can see that in this asymmetric reconstruction also. It's this other side, this distal pole that seems to be distorted. And here's the reconstruction. This is the wild type immature Kuhn virus. And then this is the immature Kuhn virus with an asymmetric reconstruction. Looks good until we get to this part right here. And you can see that there's distortion in the glycoprotein spikes. And this is a morph movie morphing the wild type or the immature wild type, the immature symmetric reconstruction with the immature asymmetric reconstruction. You can see that this region right here is distorted and probably lacks density attributable to the glycoproteins. I should also point out that it's not exactly on the five-fold vertex. This is a five-fold vertex right here. It's a little bit off from that five-fold vertex. Is it true in other flaviviruses? We looked at Zika virus. We didn't have to do any new experiments other than to reprocess the data, not imposing icosahedral symmetry. And we find that Zika is also the same observation when we do this asymmetric reconstruction. What about the mature virus? Does everything kind of fix itself as it undergoes maturation? Here is the icosahedral averaged mature virus, and here we have it with no icosahedral averaging. And we can see that we have the same kind of problem, that there is one pole, one side of the virus particle that is lacking in density. And so let me just summarize, we're still pursuing this and trying to understand what's going on. But what we'd like to suggest is that here's the RNA and capsid that forms a core on the inside in the cytoplasm. It buds up against the lumen of the ER, in which there are preformed spike proteins sitting there waiting to be grabbed. There is envelopment around this core RNA complex of these spike proteins and the envelope. And what's going to happen is that as we approach the final pinching, the neck narrows. And when the neck narrows, there's steric clashes between these glycoproteins. And that steric clash prevents the correct number of glycoproteins to be incorporated into the particle. There should be 180 E proteins in the particle, but we suggest that because of this pinching and the steric constraints that are imposed by a budding membrane and proteins on opposing sides of it, we think that it's imperfect. And what happens is when the virus particle buds, it's missing a few of those spike proteins. So let me just summarize this asymmetric reconstruction portion. The core is eccentrically positioned in the immature viruses, in contrast to the mature viruses. Okay, so it's sitting on one side, it's favored on one side and not the other, kind of pushing its way, if you will, into the membrane and causing the membrane to bud. There are deviations from icosahedral symmetry in the glycoprotein shell. These asymmetric features are possible consequences, as I've suggested, but not proven, of the assembly and budding process. But we do think that the particle has less than the ideal number required for icosahedral symmetry, less than 180 copies. If we're missing some of these E proteins, that may allow for greater flexibility of the particle and supporting some of the ideas around dynamics that I talked about earlier, because the particle is not so densely constrained. And then finally, and Michael was very keen on this from his work with bacteriophage, thinking that this distorted end of the virus particle may be a special place to facilitate the entry process or maybe the fusion process. And the details of something like that, obviously we have not yet worked out. Okay, so let me just kind of spend the last few minutes just saying a few words about antibodies, where they go and what are some of their properties. We and others, although we've done quite a number of these, have been looking at how neutralizing monoclonal antibodies bind to flaviviruses. And this is a variety of flaviviruses. These are just FAB fragments, but they're binding in different places. They're binding with different stoichiometry. And we've learned an awful lot about how they affect or how they neutralize, the mechanisms of neutralization. And we know that there are different classes binding to different regions. I'll say that in the case of dengue in particular, and Zika, some of the most potent human neutralizing antibodies are ones that cross the E protein boundaries, so that they are quaternary antibodies, recognizing pieces of one monomer and pieces of another monomer, and thereby locking these E proteins in place so that they cannot undergo the rearrangement that will be required when they undergo what I left out before, low pH mediated fusion, in which these E proteins form homotrimers. Now those, all of those images that I just showed you are studies in which we've been looking at potent neutralizing antibodies against the E protein. And I didn't clearly tell you, but in the mature virus, what's left after cleavage of PRM, the M protein sits underneath E and is really, as far as we can tell, not very accessible or not accessible to the immune selection. And so do you make antibodies against M? That I do not know. But you can make antibodies against PRM, as I mentioned before. And so this is the set of experiments that we've wrapped up now, a collaboration with Ted Pearson's group at NIAID and Michael Diamond at Washington University, in which we have antibodies against PRM, which are neutralizing. They don't completely neutralize virus particles, but they partially neutralize. And that's shown in this panel over here. These antibodies were raised against the dengue 2 PRM. And if you recall, dengue 2 is probably the worst flavivirus for completion of PRM cleavage. And what we find is that a series of antibodies are able to neutralize up until about 50%. So that's kind of consistent with that data that I presented, which was generated many years earlier on measuring the PRM content of dengue 2 virus particles. And we can show that if you influence the cleavage in a variety of ways, either by overexpressing furin in a viro cell line, or this is just the regular data that we have over here, or that you inhibit the furin cleavage, you can see that you can get nearly all of it then neutralized. Neutralization is also dependent upon the cell type. So there are a number of suggestions about the mechanism by which you're neutralizing against PRM. And I'm not going to go into that right now unless there's a question about that. But we did a pair of reconstructions against 2 antibodies that are potently neutralizing PRM12 and PRM13. They are shown here, and I'm going to show a few more slides to compare them. The resolution of these is modest. It's only about 10 angstroms for both of those. But you can clearly see that they're binding to they're binding in slightly different orientations. I will tell you that they're binding essentially to the same epitope on the surface of PRM, the very distal tip of PRM as it sits on top of that spike that I mentioned. Here is PRM12 looking down a 5-fold and a 2-fold. The 5-fold, I'd like you to look at how that fab is coming off the surface, because I'm going to compare it now with PRM13. So you can see that PRM13, rather than coming like straight off the top, is at a substantial angle relative to the other antibody. And this is the fitting of the FAB fragment, the structure of the FAB fragment into the density for the virus. You can see that PR is shown here in purple, and then we have our 3 domains in red, yellow, and blue for the E protein. And it may not be apparent here exactly the angle that we have, but what I'd like to just do is tell you that they interact and they interface with just about the same set of amino acids, but the contact for the antibodies is quite different. So that in this one, PRM12, this residue K26 is a very important residue. K26 is conserved, highly conserved in all of the flaviviruses. And we clearly are making a nice set of contacts around these residues right here. On the other hand, PRM13, K26 does not seem to be very important. And more importantly, it looks like the light chain is not making any significant contributions for binding to PRM in the PRM13 interaction. So let me just summarize that then. Characterization of a small panel of PRM reactive dengue antibodies revealed cell type and maturation state dependent differences in neutralization potency. We have a role for K26. I didn't say anything about E28 to antibody binding, and that was done through functional studies that I didn't describe and biochemical studies. These cryo-EM structures bound to immature dengue identify 2 distinct modes of attachment. And importantly, the reason why we're studying these antibodies is that if you engineer the antibody so it no longer is able to interact with the FC receptor, and you go into a mouse model of dengue pathogenesis, you can find that these antibodies are able to protect partially against antibody-dependent enhancement. And so we think that this may be some evidence that PRM and antibody, the presence of PRM, and therefore the antibodies against PRM may play a role in pathogenesis in the human disease that is the more severe form for dengue virus. So, why do we want to study these virus antibody complexes? We'd like to understand, how many molecules of antibody are required to neutralize a virus particle. We're looking at a very sanitized system in which we have, a fab fragment. It's a monoclonal. We're looking at binding to a perfect looking virus. What determines that stoichiometry of binding? What about the affinity and the potency? The neutralization mechanisms, we like to really understand. There are multiple types of neutralization. Antibody cross-reactivity. Why do some antibodies against dengue virus cross-react with Zika virus? And how does that play into disease? And how does that play into whether vaccines are going to be successful? Finally, what are we going to do with the structural data? Well, we're working to re-engineer some of these antibodies so that they have new properties, new affinities, new specificities, and new functions. And so with that, let me just conclude by saying, here's a little taste of some of the stuff that we're working on right now that needs to be refined. But these are 3 human monoclonal antibodies against Zika virus. And you can see very clearly how different their binding patterns are, how different the stoichiometry of FAB to virus is. And so we're hopeful that understanding this in greater detail is going to give us insight into how antibodies neutralize viruses, as well as being able to design new therapeutic antibodies and new generation vaccines. So with that, let me just conclude. Let me give my acknowledgement to Michael, who has, as I said, worked with me for more than 25 years before he passed away. He was an unbelievable person and a fantastic collaborator. And then I've had great collaborations with my good friend and colleague, Ted Pearson from the NIAID. Kim Dowd, a research scientist in his group, has been the lead on a lot of the studies that I mentioned, not the least of which is this PRM 12 and 13, in which we are finalizing the manuscript right now. And that's also a collaboration with Mike Diamond at WashU and David Fremont at WashU. And then finally, a lot of the antibody work has been done in collaboration with Jim Crow, who is at Vanderbilt. And then in my laboratory, Madhu Savana, who is a research scientist, along with Thomas Close, another research scientist, have been instrumental in all of the cryo-EM work that we've been doing, especially with the antibodies. Matt Thurkelson was a very unconventional graduate student in terms of his thinking, going against what Michael Rossman told him about looking at symmetric versus asymmetric reconstructions. And so that's the work that he did. Andy Miller has been doing a lot of the support work, but also more recently, Andy has a new structure for yellow fever virus and a number of other very interesting antibody studies that we've been doing. And then Devika Sarohi is a postdoc in the lab, and she was responsible for the Zika virus and more recently with the PRM 12 and PRM 13 antibody work. And with that, I will stop and be happy to take any questions that anybody might have.
Thank you very much, Richard. We have some questions that are on the chat here. Oh, there's one from Bert Semler.
Uh-oh.
Is there a function for the leftover PR domain after PRM is cleaved during maturation? Does the virus use inefficient cleavage to control the levels of PR?
Yeah. So we did some experiments with the mass spectrometry years ago that said that there is some residual PR that remains on the virus particle even after cleavage. Okay, so that's one thing. And that may play into the antibody work that I kind of ended with, right? That's one thing. Whether the PR, the released PR, has a function, I don't know if anybody has really addressed that. It's a very interesting question, considering that the different flaviviruses do it differently. So.
Yeah, and then Yang Sheng has a question about how do the temperature and pH sensitivities affect the virus behavior in the reservoir versus the host?
That's a great question. So my colleague, Michael Rossman, a true genius of structural biology. Michael loved to think more biological, but Michael didn't have as much kind of biological training. So Michael would so frequently make intuitive jumps that were based on what he thought. And so this is a perfect example of that. And you know, I jumped on it too. When we first got this structure of dengue 2, that if you incubate it at room temperature, it's smooth. If you incubate it at 37 degrees, it's bumpy. It actually goes through a transition right around 33 degrees. And so Michael said, mosquitoes, they live at low temperature. Humans are at 37 degrees and higher during viremia. We have two virus states depending upon the host. And that sounded so great. I was so excited about it. But when we looked at the other dengue viruses, it was only dengue 2 that had this specific property. They all have dynamic properties, but we can only capture these two states with dengue 2. So it's a great question, and there may be more to it, but that's what I have to say right now.
All right, and then Paul Gershon has a question. I may have missed this. You mentioned that the point of asymmetry in EM density was offset from the five-fold symmetry axis. Is it consistently offset by the same amount and in the same manner, or is it randomly offset from particle to particle? And what you showed was just one example.
Yeah, no, as far as we can tell, it's offset by the same amount independent of the virus that we've looked at. And so, again, we think that that's a physical constraint imposed when the virus particle is budding. That just a relationship, because remember, these envelope proteins that are packing early on, they've already kind of arranged in the correct icosahedral arrangement. At least we think so. So we think that by the time you get down to that narrow neck, there is physical constraints, and it's always going to be the same way as you try to fill that icosahedral particle.
And then Don Forthall asks, if you took dengue virus straight out of an infected person or animal, would you see the same sort of shape shifting, or would there be more uniformity?
Good question. It's tough to do. The data that exists suggests that there is PRM. So you do see, for example, the PRM content is high in dengue too, and so we expect it to be the same. And the fact that you have antibodies in patients that have PRM suggests that. The fact that you can raise antibodies that don't recognize that smooth structure also suggests that those antibodies are seeing a different structure inside the human or the animal.
And then this person, Rashika Pereira, I don't know who she is, but she asks, given the differences between dengue 2 and dengue 4 in maturation, do you think these different serotypes would have differences in the envelope lipid composition that would drive differences in the structural dynamics of the glycoproteins?
Ooh, well, we don't, what we know is simply the efficiencies of cleavage. We don't know how that's related or how that impacts other aspects. If you're asking a more difficult question of, do they bud in slightly different environments based on lipid content? I can't answer that, but that certainly, that may influence all of what's being cleaved.
Richard, I had a couple of follow-up questions. One is in the distorted particle, is the M protein more accessible?
So yeah, the distorted particle. So remember, the reason why we use icosahedral averaging is because we get 60-fold redundant data, okay? So the resolution of those icosahedrally averaged particles are much better. And that was one of the reviewers' complaints with us, is that, oh, you got such low resolution, what can you really say? The low resolution is because the particles are not homogeneous and the reconstruction's not that great. So the answer to your question is we can't see that.
Right, okay, yeah. And then just one more thing. For that distorted particle story, is it cell line dependent? Do you see different sort of levels of that when you grow the virus in different cell lines?
Yeah, we have not tried that. It's a good.
You might imagine that, different.
Yeah, it's a good point. I mean, to look between mosquito, I don't know, maybe, I'm thinking that maybe we did look between mosquito and Vero cells, and that there was nothing significant. I'll check on that. But that's a real good, that would have been something I would have asked right away.
Yeah.
So I probably did.
All right, any more questions? There's some people on this thing that want to talk to you, Richard,
Okay.
including our graduate students and somebody